to enhance sensitive mesenchymal interaction or hair follicle bioengineering.
Okay, good afternoon, ladies and gentlemen. I'd like to summarize the current strategy to enhance epithelial and mesenchymal interactions, EMI, for human hair follicle bioengineering. I would like to focus on the human hair follicle bioengineering, and I declare no conflict of interest. Just as a backup, but the EMI is key in hair follicle morphogenesis and cycling. So if you, in case of, this summarizes the hair follicle morphogenesis, and this is a cycling, but represented by the blue arrow here, the intensive and the cooperative EMI enables the hair follicle to be formed or regenerate itself during the life of during our life. And here I listed the representative hair reconstitution assay. Like this is a chamber assay in which you combined the epithelial cell component with the trichogenic dermal cells. In most cases, we adapt the neonatal mouse fibroblasts and grafted into the silicone chamber on the transplanted onto the in most cases back of the new mice, and then you got the hair follicle structure like this. And in this case, the patch assay, where you call a mixture both epithelial and the mesenchymal components and grafted into the subcutaneous space of the mice, in most cases the immunodeficient mice, and you got the hair structure like this. And as is presented in the last presentation, there is an assay called the sandwich assay, in which you enzymatically dissociate the epithelial dermal components and implant the trichogenic dermal cell component and graft this composite into the subcutaneous, in most cases subcutaneous space or under the kidney capsule of the immunodeficient mice. So if you take a look at those assays, we can say that there are three components essential to bioengineer hair follicles. One is epithelial cell component, and ideally it should have the capacity to respond to the hair inductive dermal signal. And other one is dermal cells with potent trichogenic activities, like as Claire says in the last presentation, the fibroblasts won't induce hair follicles, but while the neonatal dermal cells or the DP cells, in case of mice and rat, has a strong hair inductive capacity. Epithelial, dermal. And other components that we sometimes do not lay stress on or pay attention to is a microenvironment. So I'd like to go through all these components in my presentation. So first, let's talk about the epithelial components. The expected property for epithelial components for hair follicle bioengineering would be the high proliferative capacity, because to bioengineer sufficient number of hair follicles, obviously you need a large number of the cells, and receptivity to the dermal signal in other words plasticity. So this is the data from my laboratory, but if you combine the human adult normal keratinocyte with the trichogenic dermal cell components, you kind of like get hair follicle-like or hair shaft-like incomplete structures. But it's been described or reported by other investigators that this reconstitution capacity could be more enhanced if you combined the epithelial components derived from neonatal mice, neonatal human, or fetus human keratinocytes, suggesting that more juvenile or innate epithelial components are receptive to the dermal signal. So receptivity really matters in hair follicle bioengineering. As we all know, that level retaining cell technique detects the epithelial hair follicle stem cell in the bulge region of the hair follicle. And in case of mice, the elegant transgenic approaches like a GFP leveling or using the cell surface marker CD34, enabling the isolation of the hair follicle bulge cells, living bulge cells. And when you combine the bulge cells with the hair inductive mesenchymal stem cells, as you can see here, labeled by the GFP green staining, that bulge cell more efficiently, actually bulge cells are highly proliferative, and the bulge cell highly efficiently reconstitute not only the hair follicle structure but also the sebaceous gland and epidermis, suggesting that the bulge stem cell would be a nice candidate, optimal material for the human hair follicle bioengineering. In case of human, as I introduced you in yesterday's my presentation, that by using the cell surface marker represented by CD200, you can enrich the bulge cell components, and the isolated cells have a high proliferative capacity. And if you compare with control comparisons, and these cell population can be used for the hair follicle bioengineering if you can successfully collect a larger number of those cells. But it is sometimes difficult to collect a sufficient number of those cells, and if you put them in a culture, they will lose their capacity or to respond to the dermal signal or receptivity to the hair inductive signals. But we can have the better material that the group in the University of Pennsylvania recently successfully reported that by using the cell surface marker, the CD200 and the alpha-6 integrin, they can enrich the keratinocyte cell population equivalent to the human bulge cells from the human-induced pluripotent stem cells. So those cell population may be used to bioengineer human hair follicle in future. But the more tackling is that the preparation of the hair inductive mesenchymal stem cell components, and that even today, most investigators, including us, still use the neonatal mouse fibroblasts or cultured mouse dermal cells to induce the human mouse hybrid hair follicle structure. And what is the expected properties for mesenchymal components for hair follicle induction, hair follicle regeneration? One is, of course, as I mentioned before, the high proliferative capacity needs to be secured to obtain the sufficient number of those cells. And the other point is that the potent hair inductive signal, of course. So what are the possible approaches to prepare the hair inductive dermal cells? And as you can see here, in chamber assay, if you combine the low passage mice dermal cells and combine them with epithelial components, the hair follicle efficiency is great. While the high passage DPCs, the efficiency will be significantly impaired. So one possible approach is maybe expand dermal cells without losing their hair inductive capacity, which seems to be tackling. And the other approach will be use of other dermal cell population for the generation of hair inductive mesenchymal. So this is a result of our laboratory, and as introduced in the last presentation, but if you make the cell aggregate from the isolate or dissociate the dermal papilla cells, you can restore, at least partially restore, the hair inductive capacity. But there are several ways to generate cell aggregates. Like in the last presentation, the investigator used the hanging drop culture where you generate the hanging drop, and using the gravity, you generate the cell aggregates. In this case, we adopted the forced cell aggregation where you plated, you prepare the non-cell binding plates with a round bottom and put the dermal papilla cells, and with just gently centrifuge those plates, and then you get the cell aggregate like this. And this is alternative approach where we take advantage of the self-aggregated nature of the human dermal papilla cells. In this assay, we attempted the 3D culture, and we put the dermal papilla cells in the matrigel and make the self-dermal papilla cell aggregates. And these are the gene expression profile. But what I would like to say is that the orange bar represents the gene expression profile of this DP spheres, and the violet bar represents the gene expression profile of the DP sphere generated by this methodology. So both methods, so if you make these are the cultured dermal papilla cells. So obviously, if you make cell aggregates, you can get the upregulation of the DP biomarkers, but the effect itself seems to be a little bit different. So we need to be further investigate what is the optimal way to regenerate the DP spheres. And other approaches, like the investigator in Taiwan used the polyethylene covalent alcohol or synthetic material to form the dermal papilla cells, and they can get the DP, hair inductive DP cell aggregates. So the bottom line is that if you get the cell aggregates, you can partially restore at least the hair inductive capacity. But what is the optimal method to do that remains to be elucidated. Other potential approach is like addition of the activators of the signaling pathways, which seems to be lost during the culture. In this case, the mouse dermal papilla cells, the researchers at the BMP6 to the passaged dermal papilla cells, and they restore the hair inductive capacities as demonstrated like here. And in this case, we make the combination of the Wnt activators, BMP activators, and fibroblast growth factors, and supplement them into the conventional DP culture condition. And you can see that the DP cells treated with this condition restore the alkali phosphatase, which is a DP biomarker expression in vitro. So these approaches may be combined to the DP spheroid assay to to enhance the efficiency of the hair reconstitution capacity. And other possible approaches is use the other cell population, and for this, I think still the sufficient investigation has not yet to be done. And what are the potential candidates for these alternative? So sources are, for example, the dermal C cell, DS cells, and you can culture them. Or, as I mentioned in my presentation yesterday, the human iPS cell maybe, and neural crest cells, or other cell population like mesenchymal stem cell, or SKPs, skin-derived precursor cells, or nesting positive cell can be used as the alternative cell source for regenerate the human dermal papilla equivalent for hair follicle bioengineering. And at last, I'd like to discuss about the importance of the microenvironment. So microenvironment actually plays a very important role. And how we can moderate the microenvironment. One approach is positioning of the epithelial and the mesenchymal cell subsets. In other words, cell compartmentalization. So if they place them in a compact situation, maybe the crosstalk will be enhanced. And the other possible approach is support from surrounding cells. So in this case, the investigator from Japan combined the epithelial cell components and the mesenchymal components and put them in a very compact situation to make the what they call the hair jam. And since they are packed in a very tight anatomical locations, so they can communicate with each other very efficiently and induce success. And the investigators successfully obtained the hair follicle structure as indicated by here. And recently, the George Contreras group reported that the FGF9 secreted by the dermal T cell as well as the fibroblast enables the hair follicle neurogenesis in mice. So those observations clearly indicates that the environmental factor really matters. But in case of human, the number of the cells are very limited. So in my laboratory, we established what we call the hair seed assay, in which the very small silicon seed, you see the keratinocytes and dermal cells and fibroblasts, and grafted into the subcute of the immunodeficient mice, and you can get the seed structure like this. And within it, we can observe the hair follicle structure like this. Although this assay is still ongoing and we need to further improve it, but these assays can provide a useful tool to further dissect the EMI in hair follicle bioengineering. So in conclusion, efficient and stable bioengineering in human hair follicles could be enabled by combining various approaches to enhance EMI. And at last, I'd like to thank all my mentors and the lab members who contributed to this talk. Thank you very much.
Thank you very much. The nice talk. This window is now open for questions. Can I perhaps start off? Both yourself and Claire also alluded to the epithelium has to be receptive.
Mm-hmm.
Have you, or does anybody know what it is that makes the epithelium, these keratinocytes, receptive to inductive signals from the
Well, actually, this is a little bit conceptual. I frankly need to agree, but what we know is that at least in mice experiments, if you compare the non-bulge cells with the bulge cells, the hair reconstitution efficiency is much higher in the bulge cells. So maybe we can rephrase it to the stemness, maybe, or yeah.
And has anybody ever done these experiments using immortalized keratinocytes, like interts or handcaps? I mean,
I can't really remember, but I think maybe the answer is yes. Yeah, but I'm sorry, I can't really remember the result.
Very nice work.
Thanks.
I want to know whether you consider the source of the keratinocytes in terms of the body side, in terms of their ability to...
Oh, you mean the interfollicular and follicular keratinocyte from scalp?
Your source probably is foreskin, and normally they do not generate the same.
I think that is a good question because in our pilot study, the proliferative capacity will be significantly different from the scalp keratinocytes and the keratinocyte from other body sites. So I don't have the definitive answer for that question, but maybe that matters. Thank you.
Okay, thank you. If there are no more questions, we'll move on to the next speaker.
Thank you very much.
Thank you.